Comes to us from UCLA. Clarice is originally from Sao Paulo and studied a number of places, including Sao Paulo, the École Polytechnique in Paris, Cambridge, and then went to do her PhD at MIT, first in David Corey's group, and then with Paola Capellaro, and she started her group. So she comes to us with a wealth of the sort of spin expertise that many of you are familiar with from those groups. And her interest is in what quantum means for biology, which, as many of you know, has become a controversial topic. And Clarice has written a bunch of opinion pieces and editorials that you might want to take a look at. She'll probably give you the citations, promoting why this is something important to think about. And I hope you're all as excited as I am to hear directly from her about what this means. So please join me in welcoming Clarice.
You are right. No? Yeah, great. Awesome. Sorry. And why isn't that working anymore? Okay, got it. Now it will. Oh. Okay. Okay, now it goes. I'm going to talk to you a little bit about the data that exists that has been taken for the past 40 years. And then I'm going to tell you a bit about the problem with the data and how I think we can break clean with the way experiments are done. And I'm going to argue that we might be able to build quantum instruments, bona fide quantum instruments, to study this phenomenon in biology. And finally, I'm going to motivate that this is not a basic science curiosity. So our very real goal is to say, within a decade, produce what I'm calling electromagnetic interventions that are endogenous, that is, you don't need genetic engineering, non-chemical, everything is electromagnetic, non-invasive, you don't need to cut stuff around, cheap, portable, really magnetic field devices, or even a magnetic field profiles that can be applied by anyone holding a cell phone. And I'm going, believe me or not, we're going to talk from theranostics to things like space travel. So it's going to get very funky very fast. But again, don't get me, first of all, here's the people who are really the unsung heroes of these stories that I'm going to tell you today. In red, you see my wonderful postdocs, in green, my grad students, and all in blue, all our undergrads who have rotated throughout the years in our lab. So again, I do identify myself as a quantum engineer. So this means that I build instruments to study and control things that are so small and so well protected from their environment that they're better described by the laws of quantum mechanics, as opposed to laws of classical mechanics that rule everything around us. So people do get surprised when I say that I study things that might be happening inside birds and actually inside all our cells. And that's because I'm a quantum engineer interested in how quantum physics informs biology at the nanoscale. So before I explain where I'm going, let me tell you where I come from, really 2 seconds. So I am trained in the field some of you might know as quantum sensing, okay? And during my PhD, I built an instrument to do magnetometry with nitrogen vacancy centers in diamond. And the idea is that you can mathematically prove that if you use a quantum object as a sensor, your measurement is improved. In other words, the sensor quantumness enhances the measurement. And again, during my PhD, I'm not going to talk about it today, but I did work with a technological quantum sensor that works at room temperature and in the messy environment of the diamond crystal, or even the environment whose magnetic fields the electron in diamond is supposed to help sense, okay? So quantum sensors that work at room temperature under noisy environments are known to physicists. So, but my talk today starts here, actually. It starts in the chemistry. And it turns out that there's no doubt that electron spins inside proteins in solutions at room temperature are performing a type of quantum sensing. Chemists don't call it this way, but I hope you will see, for those of you who know a little bit how this type of quantum sensing works, the analogies with what's undoubtedly happening in chemistry, in the chemistry of compounds in solution. Here's where my talk starts. And finally, I'm going to tell you about the evidence that is incomplete, but it's very suggestive that electron spins inside proteins, inside cells, might be themselves be working as bona fide quantum sensors. Okay? So again, we're going to start in the chemistry. So chemists have known for more than 40 years that weak magnetic fields can alter the final products of a class of chemical reactions that depend on electron spin. Okay, not nuclear spin, electron spins. Here's how this happens. There's a chemical reaction that's happening, and at some point, the chemical reaction comes to a crossroads. At that point, the chemical reaction is actually going to look for the spin state of a particular electron and say, if the spin is down, the chemical reaction is going to continue to one branch, yielding the products that I marked there as yellow products. If the particular spin that the chemical reaction cares about is up, the chemical reaction is going to continue through another branch, yielding red products are as marked there. There's no doubt that this happens for a broad class of chemical reactions. Now, in the presence of a magnetic field, you're an audience of mainly physicists, so I don't think you will be surprised, but magnetic fields can actually put spins into superposition states. Okay, so sometimes I have to explain this to people who have never talked about spins. So the better analogy that I could come up with was to say that a superposition state is like a dimmer switch in which the electron is both up and down at the same time. But we all know that everything that starts quantum dies classical, so that after a short time, the electron is going to be better described by the laws of classical mechanics, again, behaving as a flip switch.
Just ask two questions about this. One is, does it require the system to already be oriented so that it knows what direction of magnetic field to be looking at, or is it?
No, the model predicts dependencies on the magnitude, the frequency, and for molecules that are attached to membranes that have a particular orientation on the direction of the field.
Okay, so we're talking about AC magnetic fields.
No, not necessarily. DC fields.
When you say weak, roughly what's the order of magnitude?
I'm going to get there in a second. That's the coolest part of this whole shebang, I think. Okay, so weak magnetic fields can actually put spins into a superposition state. For example, blue magnetic field can put the spin in a certain superposition state, so that if you have many molecules undergoing this reaction and the many molecules have their spins in the superposition state, it shouldn't come as a surprise that on average, the final products in a branch where you have this type of spin superposition is going to be a combination of products coming from only up and only down in a proportion that is related to the superposition character, to how much the dimmer switches to one side or the other. And a slightly different magnetic field, green magnetic field, is going to put the dimmer switch in another position so that your final products are also going to be a combination of products of only up and down, but in a slightly different proportion. So all that I've told you is correct. There's nothing wrong about it, except that when we talk about magnetosensitivity in biocompounds, this is usually what's meant, okay? In fact, the chemical reaction actually cares about the spin state of two electrons that can either be in a singlet state or in a triplet state. Here, just for ease of the argument, up-down is a bona fide singlet state, up-down minus down-up, and this guy here is meant as the triplet zero state, that is up-down plus down up. This is what those things mean. But everything else is the same. The chemical reaction actually cares about the spin state of those two electrons. Now, some observations. Okay, first observation is that, well, even though these interactions with magnetic fields are very, very brief, because they occur at a cross point, at a crossroads in the road of this chemical reaction, the consequences of this very brief interaction are felt at much, much longer timescales, when the two spins are already in the flip switch state, when all quantum coherence is lost, when the superposition is no longer living. Second point, and that's probably the most sophisticated argument that I'm going to make here, because that's the cool part. For this type of chemical reaction, I'm going to explain to you why it's counterintuitive. The only kinds of magnetic fields that can actually alter this chemical equilibrium are relatively small. Okay, here's a unusual effect whose magnitude does not only go up, does not only go down, it goes up and down as a function of magnetic field strength. Here's what I mean by that. I mean that if you have two very big fields, in the end, you have the same products. From a certain field magnitude onwards, changing this big field is not going to change the final products of these chemical reactions. That sounds weird, right? Have you seen those things before? I had not. So here's why. This whole shebang only works because and while those two electron spins see slightly different local magnetic field environments. Now, electron spins are coupled to nearby nuclear spins. The magnetic field produced by nuclear spins, as seen by the electron spin, is usually referred to as a hyperfine interaction. Okay, so in this example here, the electron spin that we care about only see a couple of nuclear spins. It's not super strongly hyperfine coupled, whereas this guy here sees a slightly larger hyperfine interaction. Well, we all know that in the presence of a magnetic field, spins will Larmor process around this field with a frequency that depends on the magnitude of the field that they see. So in this case, the leftmost spin is Larmor processing very slowly, whereas the rightmost spin is processing a little bit faster. Now, just for the sake of the argument, let's forget about the hyperfine coupling of this guy to the left. And let's see what happens when we apply an external magnetic field here in blue, okay? In the presence of an external magnetic field, well, this guy will Larmor process according to the field because that's the only thing that it sees, whereas this guy here will Larmor process with a frequency that depends on those two fields that it interacts with. Now, let's go to some cases of interest. The first thing that I would like us to consider is the case where the external field is small, in particular when it's very small as compared to the hyperfine interactions, okay? In this case, well, the leftmost spin is going to start Larmor processing with the frequency given by that field, whereas this guy here will almost not care, in that this tiny little field, I mean, it couldn't care less because mostly its frequency is related to the frequency coming from its hyperfine interactions. Now, if we wobble a little bit this tiny field, well, this guy here will care, right? Because it cares about any field that it sees. Whereas this guy here will almost not care, in that this wobbling here is very, very tiny compared to the Larmor frequency that it had. So that a tiny wobble in this tiny magnetic field will create a, if you will, a phase difference in the precession of the leftmost spin. I like to see it as the quantum sensing of the magnetic field as done by this leftmost spin. In other words, if you have taken quantum before, this is, if you will, the two-spin version of Ramsey interferometry. You might have seen that in Ramsey interferometry, you take a qubit, a spin, put it in a superposition of up and down, and the idea is that up interacts with the field differently then down, you create a relative phase between the evolution of those two states, and this relative phase is experimentally mapped into a different probability of you finding the spin as up and down that goes as a function of the field. This is exactly the same. If I had to put it in bras and cats, it would look like this. A tiny magnetic field, put the spins into a superposition of singlet and triplet. A tiny wobble of this tiny magnetic field is going to create a relative phase between those two states because of the work that this guy is doing. The rightmost guy is not, it doesn't care really about this tiny field at all. So that experimentally, this means that the probability of us finding the two spins in a triplet or a singlet state is going to be modulated by a quantity that depends on this tiny delta in magnetic field. So if I could write here the final product of the chemical reaction, here are the products for the triplet state, here are the products from the singlet state. The products from a certain superposition is going to be a mixture of those two products that is going to be modulated by this tiny change in magnetic field. Yes. So, but it's the phase difference as before, right? So before you had C here, and you had like small blue and big yellow. And now you have blue plus delta blue and big yellow. Maybe let me try to tell you if I can convince you what happens in the other regime when there's a very, very large magnetic field that actually overpowers the hyperfine interaction, okay? In this case, well, I mean, both spins are actually going to care a lot about this field, and in particular, this rightmost spin. I mean, its hyperfine interactions are going to be overpowered by this large magnetic field. So that if you wobble this magnetic field a tiny bit, neither spin are going to care. So that there is no induced phase difference once you wobble this large magnetic field. So if I could write it, I would write something like this. Big magnetic field puts the spins in a superposition state, wobble it a little bit, there is no relative phase difference between the states. So that the probability of finding the two spins in a singlet or triplet, or the probability of finding the final products as the products of triplet or the products of singlet is fixed by this large magnetic field. So that if you write the effect magnitude of the field as a function of field strength, again, it does not only go up, it does not only go down, it goes up and down. And in particular, when the external field is much, much larger, well, relatively larger than the hyperfine interactions that this guy here is seeing, the effect goes to zero. Yes?
So again, two questions. I'll come back to the order of magnitude. My problem is with the atoms we're used to thinking about, hyperfine interactions are on the order of a gigahertz, so a kilogauss.
Okay.
So you keep saying weak fields, and I'm curious
Okay.
if these molecules are much smaller.
Millitesla. Hyperfine interactions. Yeah.
Okay. The other question is, am I right that everywhere here you're so far assuming that there's no interaction between the two spins?
You don't need to.
The functions don't overlap?
You don't need to. The simplest Hamiltonian that you can write, and I'm going to tell you why in a second, is composed of Zeeman for first spin, Zeeman for second spin, and hyperfine interaction between one nucleus and one electron spin. You can add exchange terms.
Splitting, that be like...
Actually, the singlet triplet splitting is given by the hyperfine. The external, so the singlet triplet splitting is given by the hyperfine. Once you add this blue field here, this blue field is going to induce transitions between triplet 0, triplet 1, triplet 0, triplet -1. So this is quite unusual. Okay. And again, conclusion is that for two big magnetic fields, the final products are not changed anymore. Large magnetic fields do not alter the final product combination of this chemical reaction. The last point that I would like to make, and that's sort of important, I've never heard it put quite this way, is the fact that here, as in the simplest form of quantum sensing, superposition is being used as a resource. This only works because and while superposition lasts. Now, you might say, well, this guy here is entangled, this guy here is entangled. Do you need entanglement? Well, you do need entanglement, but it works because of superposition. The key point here is that superposition is causing the sensing, is enabling this sensing. So again, bona fide quantum sensing, two-spin version of your favorite diamond sensor. Yes?
This dependence on the magnetic field strength, we thought of as like the right basis field, the different magnetic fields, for example, at high magnetic field, the right basis field.
So I think what you're saying is that at low magnetic fields, there is like mixing of states. You're correct. You're correct. Yeah. Okay. Cool. Okay. So in chemistry, this has been unambiguously demonstrated at room temperature, in liquids and gases, in solids, and for fields as weak as the Earth. So your cell phone speaker is producing millitesla fields. The Earth is about microtesla. That has been shown to be active under the Earth weak magnetic field. So that's the chemistry, right? And the chemistry comes to biology because of birds. Well, I truly do not care about birds, and you're going to see how I don't care about birds. But birds have the big advantage that they brought this discussion from chemistry to biology. Here's why. When birds migrate, it's known that they use, at least as a partial cue, the tiny magnetic field of the bird, the Earth. They have a type of compass, okay? And people have been trying to understand why for a long time. And then in '78 comes this brave, crazy theoretical biophysicist called Klaus Schulten, and he makes the following outrageous hypothesis. I'll tell you why it's outrageous in a moment. He's like, well, were the same type of electron spin-dependent chemical reaction active inside cells, inside birds, birds and other organisms could sense magnetic fields to the extent that they could sense different physiological concentrations of products coming from this type of electron spin-dependent chemical reaction. Now, maybe, right, again, hypothesis, maybe a bird looking left and looking right, maybe it would see different magnetic field lines, and there's something in their eye that actually photomodulates the brightness with which they see, so that maybe they know that they should always follow the brighter image, right? Hypothesis. To date, nobody knows if, even if there is this type of electron spin-dependent chemical reaction happening inside birds, if that's truly what happens. You're an audience of physicists. Aren't you outraged by this hypothesis? Here's what this means. Okay, put explicitly. Again, I have never seen anyone put this as explicitly as this. This is outrageous because if the bird is doing that, it means that electron spin superpositions are being maintained inside the cells of the birds for long enough to function, to help as a resource, to help the birds function. Okay, we all know that quantumness does not really like warm, wet environments, right? So yeah, that's outrageous. And by how much is it outrageous? Let me tell you a little bit about this long enough. So there are better ways of estimating this, but guesstimate, okay, back of the envelope calculation. You can say that an electron senses a magnetic field if it Larmor precesses once before superpositions are lost. So for the magnetic field of the Earth, the Larmor precession takes about 750 nanoseconds. So if birds are using this mechanism to sense the magnetic field of the Earth, the final implication is that there are electron spin superpositions surviving inside the cells of the bird for at least 750 nanoseconds. More or less a tall order, right? Not completely unheard of. The nitrogen vacancy center in diamond is a magnetic field sensor that physicists use, and in its crappiest version, the naturally occurring diamond, it can retain coherences for about 2 microseconds. Again, but the nitrogen vacancy centers are special, right? So, I mean, that's still a tall order, but it's not completely crazy or unheard of by physicists. Okay, but the problem with all of this is that, well, in fact, this has never been unambiguously proven or refuted by any experiment on Earth. So if I needed to give a talk, a title to my talk, it would be like this. If they're maintained inside living cells for long enough to help with function, how can we learn to deterministically control those electron spin superpositions to tweak biology to do whatever we want? Okay? Because the thing is that in the end they fly, right? They do navigate following the magnetic field of the Earth, and it's unknown how they do so. So even though Clausius-Schulten hypothesis was and still remains outrageous, experimental biophysicists liked this idea because they were out of ideas. They tried a bunch of stuff. The birds didn't behave in a way consistent with their hypothesis. So experimental biophysicists started looking for animal proteins that could sustain those types of electron spin dependent chemical reactions. And in the 2000s, the first animal protein that was discovered to be able to sustain electron spin dependent chemical reaction was the protein there that I put there. It's called cryptochrome. Now, cryptochrome is present in the eyes of birds, in the antennas of migrating butterflies, but surprise, it's also present in all our human cells. It's expressed in all our human cells because it also has circadian rhythm regulation functions. So that protein has a chromophore, has a flavin. That means that if you shine blue light at it, the chromophore absorbs energy, gets excited, and then de-excites. And in fact, when you're jet-lagged, like I am today, and people say, like, oh, look at blue light, one of the things that you're doing is exciting your cryptochromes so that your circadian rhythm goes back to function a little bit better than mine is right now. Okay, so, and over the course of 20 years of research on cryptochrome, there's a lot of evidence that a cryptochrome might be one magnetic field sensor that is being used by nature. Unfortunately, the evidence comes at two very disconnected length scales. At chemical length scales, there is, again, zero doubt that electron spins in cryptochrome are behaving as quantum-enhanced magnetic field sensors in the way that I just explained. Let me give you more details. So it's a chemical reaction that depends on two electrons. What are those two electrons? Well, one electron is from the chromophore. It's a flavin. Okay? The second electron is thought to come from a nearby tryptophan. The way that you look at those pigments is exactly the same way that you would use to image a lot of things in the lab. You shine a laser onto a vial of this protein and then excite this protein. As it de-excites, it emits light that you can collect. If you're doing this properly for reasons that I'm going to explain in a second, you use a single photon camera. Parentheses. Just like the NV center in diamond, close parentheses, cryptochrome has a spin-dependent fluorescence intensity. This means that just by looking at how much light intensity is being emitted by your cryptochrome sample, you can actually infer if the two spins were in a singlet or in a triplet state. Open parenthesis. The reason is very similar to what happens in diamond. It's a photophysics problem. Close parenthesis, here's what happens for cryptochrome. You have there the FAD, the electron at the outer shell of the flavin that I call the FAD. It's spin up, but it could have been spin down. It doesn't really matter. That's in the ground state. There's another version of that in the excited state at much larger energies that you can access by shining blue laser. If you shine blue laser, the electron gets excited and then de-excites via fluorescence. And that you can collect. However, there's a computing process here too. Sometimes when you photoexcite the flavin electron, it decays, but it's not going to decay by emitting light. It's going to decay via loss of energy via phonon vibrations, and that's how this happens. It undergoes something that chemists call intersection crossing, whereas when it actually meets a nearby electron from the tripleton. Okay, they are in this singlet state. Actually, this is the equivalent of state preparation. Here, they always start in a singlet state for reasons that are chemical, I'm not going to talk about. But the idea is that in the presence of both an internal magnetic field from the hyperfine interaction and an external magnetic field, these two fields will put the spins into a superposition of singlet and triplet, right? And the difference energy between those two states is very, very tiny. At some point, the two electron spins are either going to separate, or the dimmer switch is going to become the flip switch, coherence is going to be lost, which is the same thing as saying that the excited electron spin from the flavin is going to de-excite back to the ground state. It doesn't emit light, it's due to vibrations, right? So if the final state of the two electron spins were in a singlet state, this guy, we're either here or here. There are two possibilities for when this de-excitation happens: when the two spins are in a singlet state, or when the two spins are in a triplet state. However, the probability that this guy here de-excites while those two electrons are in a singlet state is higher than it de-excites when the two electrons are in a triplet state, which is shown here by the width of the arrows that I plot. Okay. Here's what this means. This means that if there's one molecule undergoing this cycle many, many times, or many molecules undergoing this photophysics cycle once, there are those two processes that compete. There are three processes that compete, right? So on average, if you have many molecules, or if you repeat this process with many, many molecules, a gazillion molecules, on average, because if you are here, you go back to the excitable path again where you can emit light, on average, the spin in cryptochrome fluoresces a tiny bit more if the two spins are in a singlet state in the moment where this electron spin sort of goes back down. Okay, this difference in fluorescence is tiny, but it's measurable. So birds are not using this. They're not observing the fluorescence of cryptochrome in order to sense magnetic fields, but that's a handle that the experimentalists have in the lab in order to actually do bona fide quantum state measurements on the spins inside this protein. So in fact, if we do build an instrument that can read those tiny deltas in fluorescence, you can have, like, you can do quantum state measurement inside spins, inside proteins. Now, this is really magnetic field sensing, right? Because the magnetic field puts the dimmer switch in a certain position. The dimmer switch being in different positions, you decay back, you fluoresce at different combinations of those two states. So this is really magnetic field sensing, but there is no iron involved in it at all. Just to make sure that this is all magnetism here, or magnetic field sensing comes from spin properties. Yes?
I have to come back to my earlier question, since we're talking about a compass here.
Yeah.
Are we still just talking about sensitivity to the magnitude of the field, or is there now direction?
Here it's the magnitude, but for proteins that are not tumbling in solution, that are hooked to membranes, it also has sensitivity to magnetic field. Oh, and then you will need magnetic field anisotropy, yes. A hyperfine anisotropy. Okay. So this has been measured. by my colleagues in Oxford. So if you collect fluorescence intensity as a function of the time during which you apply light onto a vial of cryptochrome, okay? So the first thing that you see is that this fluorescence is decaying. You're killing the chromophore. But as this fluorescence is decaying, if the researchers pulse a tiny magnetic field on and off, they see a modulation in this decaying fluorescence intensity, which is a manifestation of what I just showed you. Note that the magnetic field is pulsed very slowly. This is like a steady state measurement. Nobody has taken this curve for cryptochrome, if you will, in a way that would be similar to what you would call a Rabi oscillation. This is the fluorescence. The magnetic field is pulsed very, very slowly after all coherence has decayed. So this is like steady state magnetic field sensitivity. So in the chemistry of things, it all seems to be consistent with this model. However, the next step in your in the experiments come from birds and flies and many other organisms, turtles, butterflies, that actually respond to magnetic fields in a way that is consistent with inside their cells, there being spin superpositions that drive this type of electron spin dependent chemical reaction, which, as I mentioned, has never been unambiguously verified or refuted. So can we do better? And I actually think we can. And we come to the last part of my talk, where I'm going to show you some pretty crazy stuff. Because this is not a talk about birds. This is not a talk only about cryptochromes at all. Many proteins have been shown to have magnetosensitivity. Data in cells that appear that they are in a totally cell type and organism type agnostic way to depend on weak magnetic fields in a way consistent with electron spin dependent chemical reactions actually driving the process under the hood. Okay. And again, I can flash a lot of papers that have been published over the years showing that weak magnetic fields regulate the production of reactive oxygen species, of cellular respiration, cellular glycolysis rates, DNA repair, how ion channels functions, planaria regeneration rates, human cellular stem cell pluripotency development, a bunch of stuff, right? One of the coolest ways that give you a hint that those effects are magnetic field dependent in this way is when you can calculate the magnitude of the effect as a function of field strength in a way that does not only go up, does not only goes down, it goes up and down. And there's plenty of evidence like this in the literature. I can flash as many papers as I want, but people usually get what I'm getting at when I show this slide. So this slide comes with a story. I have a friend in Munich. His name is Peter Fierlinger. He's a bona fide precision measurement physicist. What he does is he's like world leader in developing hypomagnetic chambers. And usually what he does is like he shoves ultra cold atoms experiments inside. But bio people came to him and were like, well, we want to use one of your chambers to do experiments under hypomagnetic field development of tadpoles. Shall we do it? And he was like, yeah, let's do it. And so again, your cell phone is producing millitesla fields. Earth's magnetic field is microtesla. Inside one of his chambers, you have a DC noise level of about 2 nanotesla. Okay? So first condition, and this experiment has been now reproduced by Dima Budker in mines. First condition, he applied inside the hypomagnetic chamber a mimicked magnetic field of the Earth. After 2 days, which is as long as he could go before, he would need to apply for a bio permit. He's a physicist, right? So the tadpoles looked normal. If you grow the tadpoles under the hypomagnetic field condition, like another batch, about 35% are non-viable after 2 days, which is crazy, right? So, and then you start asking all sorts of questions. Well, what's the magnetic field in Mars? Answer, it's like 1 divided by 50 the magnetic field of the Earth. Can we reproduce in Mars? Can we space farm in Mars? Okay. So nobody really knows what's going on. But because, so to recap, you don't even need to apply a magnetic field. You can be passively shielding the super tiny magnetic field of the Earth and things go crazy. Nobody really knows what's going on because it's just a picture with a 2x magnification of tadpoles. My best guess is that it's melanin. So actually there's plenty of nice spin physics in melanin, which gives the tadpoles and our skin its colors. But in any case, quantum or not, the tadpoles are sensing the magnetic field of the Earth or its absence. And the magnetic field of the Earth is very, very tiny. And again, if tadpoles are sensing the magnetic field of the Earth via this mechanism, it means that electron spin superpositions should be living inside the cells of the tadpoles for at least 750 nanoseconds. Note that if you want superpositions inside cells to see slightly larger fields like your cell phones, you don't need the spin superpositions to live for as long as that. To sense millitesla fields, you would only need spin superpositions to live for about 50 nanoseconds, which is much better than 750 nanoseconds, which is what I think might be going on in some heuristic application of magnetic fields that people have found, okay? So accidentally found weak magnetic fields are actually being commercialized to do a bunch of stuff. For example, this is from a company called Novocure. They produce those electrodes called Optune that actually can decrease proliferation rates of glioblastoma, which is like a very aggressive type of brain cancer. When you look at the magnetic field strengths and frequencies, the strengths and frequencies are consistent with what you would expect for spin physics for hyperfine couplings. So they don't really know what's going on. It's FDA approved. The company is valued at like $5 billion US, and they don't really know how it works. They say it's a combination of inflammation responses like reactive oxygen species, disruption of mitosis. So you might not know, but like when cells divide, the spindle, the chromosomes, they're pulled apart by microtubules. And I'm going to show you that even we have seen that microtubules are disturbed by weak magnetic fields. Third thing, they see that the cells in the glioblastoma have their cytoskeleton changed. We also see this. And finally, that there's a DNA repair misregulation. So it's a combination of those 4 factors, and those 4 things have been shown by different people, even by us, to be tweakable by weak magnetic fields, even by DC magnetic fields. No frequencies involved here. On this right panel, you see this guy here called Alfredo Obregon. He's a biologist who knows his spin physics, and he has not 2, but 3 startups. One that reduces breast cancer tumors, one that does muscle regeneration, and the third one that actually increases the proliferation rate of lead-grown meat using weak magnetic fields. If there's any biologist in the room, heuristically, he sees that cells that overexpress this gene called TRPC1 are more susceptible to magnetic field treatment, but nobody knows why. So, and here's the problem with all those beautiful applications, right? So people have been populating this huge phase space of magnetic field frequency, intensity, direction, exposure duration, haphazardly for like 40 years. I liken it to being in a haystack, grabbing a little bit of hay and finding a needle. And then people put a point in this phase space. You cannot link those phase space points. You cannot predict anything, right? So can we do better? I think we can. We need to break clean with this mode of taking data because we need causal data that is systematic and with predictive power. And here's how I propose to do so. Grab a microscope, put some coils, synchronize everything down to the nanoseconds because that's the time scale of spin physics. Couple that to a single photon camera. You put those 4 things together. You can call those things a glorified microscope. I call them a quantum instrument. This type of instrument is not different from the type of instrument that you would build to play with the electron spins in diamond. But here we have spins inside proteins, inside cells. You put those things together, you might get what I'm calling causal data that coupled and I'm going to show you in a second how, coupled to simulations, can give what I'm tentatively calling the code book on how to control spins in biology for function. So our scientific goal is really to learn how to tweak those degrees of freedom in biology, but technologically, again, right now this is science fiction, but where I want to be, say, in 10 years is take your phone, you have an app, and then you click your app. Well, today I need help with wound healing. You click wound healing, and then your magnetic field makes the exact magnetic field frequency and intensity that you need, and then you do something like this. Okay, so there is, it's not a basic science question at all, right? I think the knob is there. We need to learn how to tweak it, as people have been doing for a lot of time, but in a way that has no predictive power. So, and then this happened. Like, so I'm a pandemic professor. Things have not been okay in my life for the past 3 years. It's been crazy. We're very behind. But I'm going to show you where we are in building 2 of those quantum instruments. They're not ready. And I'm going to show you in a bit where we are, while we wait for things to happen around us, while we dry our lab that has been seriously flooded. So here is the blueprint of our quantum instrument. Again, 4 ingredients. We synchronize our microscopes actually with the Python control software that is used by atomic physics groups. We have a microscope. We are actually designing hypermagnetic chambers with the tadpole guy in Munich. We have coils, and we have single photon detectors. Actually, we also have a regular camera to take pictures of the cells, and the single photon camera is to do spin measurements, if you will. Okay, so we're building 2 of those microscopes. One is called a lattice light sheet, which is very good because it goes from single molecule to, say, embryos and small tissues. So we're building the excitation pathway. This is being designed, and our total infernal reflection microscope is almost ready to take data. This is a very cool water-cooled 3D magnetic field. What's the name? It's like a coil type stuff, and this is the TIRF, the total internal reflection platform. Once those experiments are ready, here's how we can use theory to go back and forth between theory and experiment. I'm going to use cryptochrome as an example, just because historically that's the most well-studied protein that can sustain electron spin-dependent chemical reactions because of birds. The field is completely focused on birds. So you take cryptochrome, you take the Flavian photophysics, you take the redox cycle of the 2 electrons involved. So even though one cannot predict which proteins are going to be magnetic field sensitive, I don't think it should come as a surprise that usually people find out that proteins that are magnetosensitive are proteins that are involved in what's called redox biology, biology where you gain or give away electrons. So you get published parameters from the photophysics and from the redox cycle, and you input them into like real open quantum system simulation. Simplest Hamiltonian is 2 electron spins that do not talk to each other. You need Zeeman interaction here, Zeeman interaction here. Third spin is a nuclear spin that is coupled to only 1 electron. With this very simple Hamiltonian, you can start making predictions. I'm going to show you one. I'm dying to take this curve experimentally. For example, here's what we see. For cryptochrome in solution, okay, tumbling around, if you plot the magnetosensitivity, how sensitive the protein is simulated to small changes in magnetic field as a function of magnetic field strength, first thing that you see from the simulations is that it goes up and down, as it should. At about 10 times the magnetic field of the Earth, the magnetosensitivity is super well reduced. And what's super cool, I think, is that it peaks very close to the magnetic field of the Earth, which is either a coincidence or there might have been some sort of evolutionary pressure if birds are truly using this in order to to navigate, that the protein works well under the field where the birds are doing it, right? We are just lowly quantum instrumentalists, but we think that people who do directed evolution of proteins can actually try to apply evolutionary pressure to this type of system. So you take this type of simulation, you go back and forth between data, and you start being able to predict, if you're here, how do you go from this magnetic field to this magnetic field? You can start connecting the points in this very large phase space with the help of this type of simulations. Now, here's what we've been doing while things have not been working around us. We've been taking, in the short term, more correlative data of magnetic field effects in biology, okay? So one of the things that we do now, so since February, we're a physics lab that can culture cells. So this is an incubator. Cells like to be inside. We can put inside the incubator, for example, a hypomagnetic chamber that is commercially available, or we also designed our own water-cooled coils. And for bio stuff, you really need to be conscious about, well, for quantum experiments too, but we know that temperature effects, if they're present, they're smaller than 0.1 Celsius. And we can do experiments both under hypomagnetic field conditions, with conditions of cells inside the incubator, and with actively applied magnetic fields. And some of the results. First of all, a null result. So some things that people do, bio people do, is something called wound healing assay. So you get a plate of cells, you scratch your plate of cells, you create a hole, and then you see how fast the hole closes. And people in the past have shown that weak magnetic fields help with a wound healing. We do not see that, and our data analysis is we press a button and we analyze the data. Nobody's counting the data. It's all very proper. We do not see a difference in the area, how fast the area is closing as a function of magnetic field. I was presenting this new result to the NIH the other day. And the NIH is the US National Institutes of Health, who are starting to become aware of this field. And someone asked, so were the cells environmentally stressed? And I'm like, what do you mean? I don't even know what you mean. And then they said, well, actually, if the cells are under the culture medium, the wound healing process is very different if the cells are with their medium, or if the cells are stressed, that is, that they don't have food. So basically, what the guy suggested was that we might see a difference in wound healing if we stress the cells. That's what we're trying right now, because in the past, people have seen different wound healings as a function of magnetic fields. We don't see it yet. What we do see in a reproducible way is that using our active coils, a magnetic field that corresponds to about half DC, half to your cell phone speakers' phones, does seem to alter cellular morphology. For example, the exposure parameters we took from the literature, but cells actin, which is like one of the filaments that constitutes the cytoskeleton of the cell, usually they're more straight. In the presence of a magnetic field, they get less straight. The contrast is not super good. The mitochondria, they appear to be in different positions of the cells. And microtubules, which is the other big component of the cell cytoskeleton, we see different polymerization rates of microtubules. That is, they don't aggregate as much in the presence of this weak magnetic field, which again might explain why people see differences in mitosis, because for mitosis, you need microtubules to pull apart the chromosomes. So with microtubules in particular, I'm learning those things. You can do an assay where you disrupt the microtubules and then try to see them come back. So there's this chemical called nocodazole that destroys the networks of microtubules, and then we see the recovery of the microtubule in the presence or in the absence of magnetic fields. And what you see is that after 2 hours, either with the magnetic field of the incubator or with this tiny magnetic field, a very strong difference in like where the microtubules are found back into the cell. And we can quantify this. I mean, it's been very hard to quantify things in a way that a physicist is happy with doing those bio experiments. So the other big result that we had has to do with reactive oxygen species that give you cellular oxidants. That is like ROS, reactive oxygen species, they're involved in signaling, but if you have too much, that becomes a problem. H2O2 is one of such species. So it's the same data if your physicists look left, if you're right, if your chemists look right. I prefer, this is a contrast between the production rate of this cellular oxidant as a function of exposure time of the magnetic field. And this is just the ratio of the production rate of that cellular oxidant for the exposed cells divided by the control cells. And this is the no effect line. So we think that at some point the cells are adapting back. There's some resilience built in, but I do stand by this point. I trust the data that for very short exposure times, there's a 40% larger rate of production of H2O2. Yeah.
So somehow your plot for the biologist says something like a 3 standard deviation effect, but the physicist version is less than 2 standard deviations. So I don't know if I stand by it. Depends which error bar I trust.
Well, for this guy, I mean, you see there are 4 quantities here. Yeah. If you do error propagation, you do have like larger error bars.
Well, I just mean that the confidence interval should exclude the no effect line to the same probability in both cases.
But how do you do error propagation? Because here you do error propagation of this divided by this. And here you do the error propagation that involves like this two, and a division. There's no way that your error bar is not going to be larger here. How do you, I'm curious.
Well, I think you're in a regime where you don't actually have a standard distribution. So you probably have to do a more complicated confidence interval calculation.
I agree. And we're trying to find better ways of.
I guess these days it's just like, but I think
no, I won't do this. I won't go there. P-value. So it's been a challenge, right? How do you present this data in a way that I'm happy with? For example, this guy here. So, that's another. Let me explain the experiment and why I'm unhappy with the way this data is plotted. So, there's this chemical called DCF. DCF is a chemical that when it's not oxidized, it does not emit light. The moment that it's oxidized, it starts fluorescing. So the idea being that, and bio people use this all the time, they drop cast DCF on top of the cells. If there are reactive oxygen species, they're going to oxidize DCF and you're going to see light coming from your sample. And what we do see is that there's more overall ROS for cells that are exposed to this weak magnetic field. And have a look at this. This is completely arbitrary units. This is the data that is spit out of the machine. Okay. So first of all, the machine adds noise in this direction. There should be no noise here, but just it plots it so that you can see the points better. This is adding a lot of noise. So how do you analyze? How does this compare to this? I don't have the idea, right? I mean, apart from showing all the points and the average, I don't know how to do a better comparison, to do a proper. We're trying to do better than what's usually found in biological experiments, and I don't know yet how to present this in a way that I'm comfortable with. Yes.
Why is it adding noise in this?
Because it's stupid. I mean, it doesn't make sense. It should be plotted on the same line. It's the same condition.
You're saying it's not actually adding them, it's just for presentation.
No, it's just for presentation.
It's just splitting the points across.
The machine gives you this, and I'm like, why? Why don't you give me a straight line?
Or is it the the distribution of the.
But then the first thing that I saw was like, well, why is there noise in this? Okay, so this, yeah, there is, I mean, there's just this condition and this condition. Okay, so again, we come to the end of my talk. I've shown you a lot of weird stuff, right? From microscopes to cells to tadpoles to simulations. And I'm going to end in the end in the way that I always do, by saying, may the quantum be with you.
Thanks again for a really stimulating talk, and we're looking for questions. I'll turn the microphone to anyone who has to ask. The recording, of course, the person on the furthest side of the room. Pass this over there, actually.
My question is very simple. Like, look, what if you grow like the tadpoles, very high magnetic field? Because you said like typically these chemical processes would depend not only on like will be poor for the low and high magnetic fields.
So at very high magnetic fields, other things might happen that for example, things that are more usually associated with NMR, right? But there's something that I didn't tell you. If you go to far enough to the right, this curve actually goes up a little bit again due to a mechanism called delta G. The two electrons have slightly different G factors, and then this curve goes a little bit up again. So, but mostly the effect of the magnetic fields in those chemical reactions are due to weak magnetic fields.
Okay.
Very weak. The other effect appears also like at Tesla levels. This is to say that weak magnetic fields you don't need a big magnet, right? Your phone is already enough to actuate on biology.
So typically biological systems are more afraid of very, very low magnetic fields than they're afraid of higher magnetic fields.
Who?
Like biological systems, like I don't know, cells or something.
The cool thing about this is that the magnetic field can modulate up and down things. So magnetic fields can do either good or bad, depending on what you want to regulate. But yeah, the magnetic fields that alter those things are weak magnetic fields.